basic concepts which are there in the 4th chapter, that is animal classification. Okay, the first one is levels of organization. Now in this levels of organization, it can be cellular level, tissue level, organ level, or organ system level. So when we'll come to various organisms, okay, one by one we'll see that some of the organisms are having only cellular level, that is only a single cell is performing all the function. In next class, okay, or in next animal, you will see that there is formation of certain tissues. In some other organisms, you will see that the tissues get in together and they form a particular organ, and these organs in next animals, these organs come together and they form an organ system. Okay, so there are various levels of organization in these animals. We will see. Okay, now the second point we can say that the digestive system. In some organisms, they will have, okay, in some organisms they will have complete digestive system and some will have incomplete digestive system. Now what does this complete and incomplete mean? When an animal is having a mouth and an anus, okay, they are having two openings, that is mouth and anus. These organisms are having, or we can say that these organisms are having what? Complete digestive system. But when these organisms are having single opening, that is only mouth or anus, okay, so these organisms are called incomplete organisms. Okay, so we'll see several complete organisms having complete digestive system, and will also come in, will see, or we will discuss characteristics of certain animals who are having incomplete digestive system. Okay, now the next one is the circulatory system. Now the circulatory system can be of two types. Okay, so one is the open type and the other is closed type. In the open type of circulatory system, as the name suggests, it is open. Okay, what does that mean? It means that the heart will pump the blood and the tissue and the cells will directly bath into the blood. Okay, now in the closed system, it will be seen that a blood is pumped from the heart and it is circulated with various arteries and veins having a particular diameter. Okay, so it is a closed circulation, that is arteries and veins will bring the blood, and in open system you will see that the cells and tissue will be directly bathed into the blood. Okay, so the circulatory system are of two types, that is open and closed. Now when we come to the next characteristics, that is the symmetry. Okay, now in symmetry you will see certain organisms which are asymmetrical. Okay, which will be the shape of these organisms will be irregular. Okay, certain organisms will show radial symmetry. Now what does that mean, radial symmetry? In radial symmetry you will see an organism, and when you try to cut that particular organism in any plane, you will get two equal halves. Just remember that when you'll make that organisms divide into two halves from any plane, okay, so the organism is radially symmetrical. Okay, now other one is bilateral symmetrical. Now bilateral symmetry is symmetry in which the organisms can be divided into two equal halves only in one plane. Okay, as we know, we humans, when I make an incision in this way, so my left and the right half will be almost same when only the outer side is observed. Okay, but when a section comes like this, so my upper half and lower half will not be same morphologically also. Okay, so bilateral symmetrical animals will be those animals which can be divided into two equal halves only in one plane. Okay, you have to remember that only in one plane. While in radial symmetry, the organism can be divided into two equal halves in any plane. Okay, like a ball. You will cut that ball in different plane and you will get two equal halves. Okay. Now, there are certain terms like the blast layers. Okay, that is ectoderm, endoderm. Okay, so when the organism is differentiating, it forms different blast layers. Okay. Now, certain organisms will have only two blast layers and an undifferentiated mesoglia layer, the middle layer. So, these organisms are called diploblastic. Diploblastic and triploblastic. Okay. We can say that in diploblastic animals, see, this is the layer, this is the single layer and this is the internal layer. Okay, so this is well differentiated and this one is well differentiated, but this internal layer is not well differentiated and this is called mesoglia. While in triploblastic animals, you will see that the outer layer is well differentiated, the inner layer is well differentiated and the middle layer is also well differentiated. Okay. So, when the middle layer is well differentiated, it is called what? It is called mesoderm. This outer one is ectoderm and this inner one is endoderm. So, when the organism possesses all the three layers, that is ectoderm, mesoderm and endoderm, these organisms are called triploblastic and these are nothing but the blast layers which are formed when the organism is differentiated, and you will come to know that various organs will be formed from these blast layers. Okay. Now, in the organisms in which this middle layer which is not differentiated well is called mesoglia, and these organisms, as they are possessing well differentiated only two layers, that is ectoderm and endoderm. So, these organisms are called what? They are called diploblastic. Okay, I have shown the pictures and I have discussed these basic terms in detail in slides also. Okay, I will explain there also. Now, the third one is the presence of coelom. Okay, a cavity. When a cavity is present between the body wall and a gut wall, okay, then this, and it is lined by mesoderm, we call that animals as coelomate animals. Okay, we will call them as coelomate animals. So, the organisms which will possess or which are having an particular cavity between the body wall and the gut wall which is lined by mesoderm is called as coelomate animals. Okay. The organisms which are not having this coelom cavity are the acoelomate animals, and there will be certain organisms in which, see, this is the outer cavity, or you can call this body wall, this is the inner one. Okay, and there will be pouches of the mesoderm. Okay, so they are looking like they are having a cavity, but this cavity is not true. Okay, and so these animals are called as pseudocoelomates. Okay, we call these animals as pseudocoelomates. Okay, pseudo means false, pseudocoelomate. Acoelomate will not have any cavity. Okay, all of the layers will be there and no cavity will be present. These are the acoelomate animals. Okay, And the third one, that is the coelomate animals, will have a structure like this. See, this will be the outer body wall. Okay, this is the inner body wall. Okay, and you'll have these particular mesoderms, mesodermal lining like this, and here also, and this will be the cavity. This will be the coelom. Okay, and so these animals, this is the cavity, these animals are called coelomate animals. Okay, so here when we were talking about the basic of, or the basic characteristics of the fourth chapter, we discussed level of organization, that was cellular, tissue, okay, organ or organ system level of organization. The second one was the digestive system, which can be complete and incomplete. Circulatory system, which can be open and closed. Symmetry, which can be of three types: asymmetrical, radial symmetry and bilateral symmetry. And presence of this middle layer, okay, that is on the differentiation on the basis of the blast layers present, that is diploblastic and triploblastic, and this is the presence of the coelom present between the outer wall, okay, that is the body wall, and the gut wall. Okay, other characteristics are also there, which I have discussed in the slide. Okay, these were some of the very important characteristics, so I made a video also, and the other characteristics and these characteristics once again I will discuss in the slides. Okay, so this is the starting of the fourth chapter, and this is a very important chapter. Here you have to remember each and every characteristic. Good morning. So this is the detailing of the basic terms or the basic concepts you need to learn for the fourth chapter, that is animal classification. I have discussed various basic and important terms in the video also, and here I have prepared this slide also for you. So the basis of classification. Bases of classification are arrangement of cells, body symmetry, nature of the coelom, patterns of digestive system, circulatory and reproductive system. Now A, first we'll start with A, that is level of organization. So organization is the structural differentiation of animal body. First is cellular level of organization, that is the cell will do all the function. In this, the cells are arranged as loose cell aggregates, example poriferans. Second one is tissue level of organization. In this, the cells performing the same functions are arranged into tissues like coelentrates and ctenophores. Third one is organ level of organization. Okay, in this the organs will be formed. In this type, the tissues are grouped together to form organs, and the organs are associated to form organ systems, and each system performs a specific physiological function. Example, all higher animals, from platyhelminths to chordates. Now, organ systems of different animals show complexities. Example, the digestive system of cnidarians and platyhelminths is incomplete. Okay, that is, it is having only a single opening which serves both as mouth and anus, and the second is the complete digestive system, which has two openings, mouth and anus. Now the second one is the circulatory system. The circulatory system is of two types, that is open and closed. In open type, the blood is pumped out through heart and cells and tissue are directly bathed in it. And in closed system, the blood is circulated through vessels, which are the arteries and the veins. Okay. Next is the body plan. Animals generally have three types of body plans: cell aggregate plan, blind sac plan, and tube within a tube plan. Okay. So in cell aggregate plan, the body consists of aggregation of cells, and it is usually found in sponge. Blind sac plan is the plan in which the body has a single cavity with one opening to the outside. Two single opening act as both mouth and anus. And the third is tube within a tube plan. That is, in this the body has two tubes, one formed by the body wall and the other formed within it by the digestive tract. Okay. Now the third is the body symmetry. Okay, so the animals can be asymmetrical and symmetrical. So asymmetrical animals are the animals in which the body cannot be divided into equal halves through median plane. Okay, and the simplest animals have no symmetry, like sponge and snails, etc. While when we are talking of the symmetrical animals, we will able to see two symmetry in symmetrical animals, that is radial symmetry and bilateral symmetry. In radial symmetry, body can be divided into two similar parts by any plane along boreal axis of the body. And in bilateral symmetry, body can be divided into two identical right and left halves by a section passing through the longitudinal axis. Okay. Now here, when the phylum comes, you can see or you can remember the example. No need to remember the example at this point of time. Okay. The body of bilateral symmetrical animal has an upper or the vertebral dorsal side, a lower ventral side, left and right lateral sides, anterior cephalic side and a posterior anal side. Okay. Now the fourth is the presence of the germ layers. Okay. These are the layers of gastrula from which all body organs are formed, and these are termed as germ layers. The three types of germ layers are present: ectoderm, mesoderm and endoderm. Okay, so ectoderm is the inner layer which gives rise to outer covering of an animal. Okay, mesoderm, it gives rise to muscles, skeleton, circulatory system, kidney, reproductive system, and endoderm, it gives rise to digestive tract and associated organs. Ectoderm is the outermost covering, endoderm is the innermost covering, and mesoderm is the middle layer. Now in some animals where there are only two layers, that is outer ectoderm and innoderm, and a middle layer which is not well differentiated is called mesoglia. Okay, so these type of animals are called diploblastic, and in triploblastic animals they have well developed and well differentiated all the three layers, that is outer ectoderm, middle mesoderm and inner endoderm. Okay, so these animals are called triploblastic. Now the fifth one is the body cavity, the presence of the body cavity. Now this cavity is present in the space between the body wall and the gut wall. Okay, so three type of animals are there on the presence of the cavity. Okay, the animals which are possessing true cavity are called coelomate or eucoelomate. The animals which are not having any cavity are called acoelomate, and the organisms which are having a cavity like thing but it's not actually a cavity, that is, these are called pseudocoelomate. Okay, now you have seen in amoeba we call the legs as pseudopodia, means false legs. Okay, so pseudocoelom means false cavity. So acoelomate animals have no coelom. The space between the body wall and the digestive cavity is filled with matrix, that is parenchyma. Okay, now in pseudocoelomate they have no true coelom, but the body cavity is partially surrounded by mesoderm, and mesoderm is present in scattered pouches between ectoderm and endoderm. Coelomate animals are the true coelomate or eucoelomate, in which the body cavity is completely surrounded by a mesoderm, and coelom is lined by the peritoneal layer and filled with coelomic fluid. Okay. Now the other type is the hemocoelomates. Okay, here the true coelom is reduced and it is filled with blood. Okay, hemoglobin. So we are aware of hemoglobin pigment which is present in blood. So anything related to heme is blood. So this is a coelom in which blood is filled. Okay, and what are the functions of this coelom? It will accommodate visceral organs. Okay, visceral organs will be present in this cavity, and coelomic fluid gives moist environment to the visceral organs, thereby reducing the friction, and it is also acts as a shock absorber. Okay, now when we talk about a very vital organ in the body like brain, now brain is protected with the help of skull, but skull is a solid structure. Okay, now this brain, surrounding the brain you will find three membranes in which cerebrospinal fluid will be present, and that coelomic fluid or the cavity fluid will prevent the brain from the outer abrasion with the skull. Okay, by reducing the friction from the skull. Okay, so in this way these cavities and the fluid work. Sixth one is the segmentation. Segmentation is a division or differentiation of the body into distinct proportions called segments. Okay, this is present in annelids, arthropods, and these segmentation is of two types, that is metameric segmentation, which is the true segmentation, and the body will be divided both externally and internally into number of segments. And second one is the pseudo metamerism. Okay, in this we will feel like the body is internally divided, externally divided as well as internally divided, but it will only be the external division. Internal division will not be there, so we call that as pseudo metamerism. The seventh one is the notochord. Okay, now notochord is a rod-like structure formed during embryonic development on the dorsal side. It is mesodermally derived. Animals with notochord are called chordates, and animals who do not possess this notochord are called non-chordates. Now, digestive system, we have already talked about the incomplete digestive system and the complete digestive system. So, complete means having two openings, and incomplete means having only one opening. Now, digestion can be intracellular as well as extracellular. Now, what does this means? Intracellular digestion occurs within the cell. Okay, in this the digestive enzymes are poured into the food vacuoles where digestion of food takes place. It occurs in protozoans and sponges. And in extracellular digestion, that occurs outside the cell in a cavity. In this, the digestive enzymes are poured into the cavity for digestion to take place. It occurs in higher invertebrates and all vertebrates. Okay, now this I will explain in a way. You have seen the structure of the amoeba, and in an amoeba food vacuole is present. So there is a presence of a particular vacuole in the cell itself, and the digestive enzymes are present inside that. So when the food will be engulfed, it will be digested in the vacuole itself. While when we talk about extracellular digestion, okay, we are having our digestive system. So mostly the digestion occurs in the stomach and small intestine. So it is a cavity. Okay, cell are there, but there is a cavity, there is a hollow space where the digestion will occur. Okay, so this is extracellular digestion. Now, excretion is the removal of metabolic waste from the body. Now, on the basis of this, they are monotelic animals, urotelic, uricotelic, aminotelic, and guanotelic. Okay, basic you have to learn about these: monotelic, urotelic, and uricotelic. So, these will a monotelic organism will excrete ammonia, urotelic will excrete urea, and uricotelic will excrete uric acids. Okay, so the uricotelic animals will have the excretory material, a little bit of solid. Okay, now aminotelic will excrete what? Amino acids, and guanotelic will excrete what? Guanine, a specific type of amino acid. Okay, so here you don't have to focus on all of these, but you should be aware of the names, and the first three you have to learn, that is monotelic, urotelic, and uricotelic. Okay, so we are what? We are urotelic animals, that is we excrete urea from our urine. Now, 11th one is the fertilization. Now, fertilization is nothing but the union of male and female gametes, and it is of two types. External fertilization, okay, when the fertilization occur outside the body of the female, and internal fertilization, when fertilization occur inside the female body, it is called external fertilization. Internal fertilization. When outside the female body, it is external, and when inside the female body, it's internal. Okay, inside, internal. Now the 12th one is the last one is the development. Now there are two types of development, that is direct and indirect. Now in direct development, the young ones resembles the adults in all respects except color and size, as we can see in humans. Okay, so the baby which is born is having all the basic designs which are similar to the parent, that is 2 eyes, ears, nose. Okay, and in indirect development, what will happen, that the young ones do not resemble the adults. As you can see, the larvae of mosquito is very different from the adult mosquito. Okay, so this is a type of indirect development. And as we can see that the butterfly, okay, the butterfly when formed, the larval stage and the adult stage is very different externally and internally from each other. Okay, so when these pass through different juvenile stages to when there is indirect development and when the organisms pass through various different juvenile stages for attaining the adult form, this process is called metamorphosis. This is seen in silk moth, housefly, frog and butterfly. Okay, so these are the basic concepts of the classification. Next we'll start with the characteristics of a particular phylum, and here in this chapter you have to remember all the characteristics, you have to memorize everything. Okay.